Hi everyone, this is one of the most fun topics to talk about. This is Unit 18 and we're going to talk about hot topics and infectious diseases, which are all those communicable diseases causing significant clinical illness among human populations. So stay tuned. We're going to start out with a few that you're already familiar with, but we're eventually going to be talking about some you're unfamiliar with. So let's get started. I first want to talk about MRSA, methicillin-resistant Staphylococcus aureus. Now, anyone can get MRSA, especially people that are involved with crowds, people that have skin-to-skin contact, people that share equipment and supplies. These are the kind of people that are more at risk of developing MRSA. 5% of all hospital people have MRSA. That's kind of interesting to know. And we can prevent MRSA by covering wounds, washing your hands often, not sharing things like towels and razors. Okay, now there are two types of MRSA. One is called hospital-acquired MRSA, and the other one is community-acquired MRSA. In the case of hospital-acquired cases, those MRSA cases usually involve septicemia, pneumonia, and surgical wound infections, while community-acquired cases of MRSA are usually skin infections. Usually those patients have pustules that they seem to think are spider bites, but they're actually pustules of MRSA. Now, we can screen for MRSA a couple different ways. One way we can screen for MRSA is taking our Staph aureus and putting it on a Mueller-Hinton agar with 6 micrograms per mil of oxacillin. If your bug grows on that oxacillin plate with the 6 micrograms per mil of oxacillin, that Mueller-Hinton plate, then we know it's an MRSA. Probably the most common way to test for MRSA, but this is an expensive test, is just a latex agglutination test, and that's for what's known as the PBP2A antigen. Now we have a problem because Staph aureus that is MRSA can often be treated with vancomycin intravenously. That's kind of the last line of defense to treat MRSA that are resistant to everything else. Unfortunately, now we are starting to see vanco-intermediate Staph aureus, and there have been a couple of cases of vanco-resistant Staph aureus. So these VISAs and VRSAs are diagnosed with MIC and vancomycin screening plates. Okay. The first vanco-intermediate Staph aureus was seen in 1996 in Japan, and the first few cases of vanco-resistant Staph aureus were seen in 2002 in Michigan, Pennsylvania, and New York, and I know some of those cases were catheter tips. Now what we can do is we can take our Staph aureus and put it on a brain-heart infusion agar with vancomycin, and usually that vancomycin is at a concentration of 6 mics per mil. If our organism grows on that vancomycin plate, it means that we have a VRSA, which is kind of scary. VRE is vancomycin-resistant enterococcus. It can cause things like urinary tract infections, septicemia, and wound infections. VRE is often spread by person-to-person contact with contaminated hands. Okay, VRE is responsible for 4% of nosocomial infections, and those, of course, are hospital-acquired infections. And usually the culprits for vanco-resistant enterococcus would be Enterococcus faecium and Enterococcus faecalis. Those are your most likely VRE strains. Now, if we want to test our enterococcus to see if it's a VRE, we can put it on a bile-esculin azide agar with 30 micrograms of vancomycin. If you have a zone of inhibition, it means the strain is sensitive to vanco, but no zone of inhibition means that your strain is resistant to vancomycin. Okay, we can also do a vancomycin MIC, and we can do intrinsic resistance tests, but those are just a few ways that we can help to identify vanco-resistant enterococcus. Clostridioides difficile causes diarrhea and colitis. It infects the colon and it is associated with overuse of antibiotics. It is responsible for what's known as pseudomembranous colitis and antibiotic-associated colitis, and this can actually be life-threatening. Okay. Practitioners overprescribe antibiotics when 30 to 50% of hospitalized patients really don't need them. It is the overprescribing of antibiotics that seems to lead to Clostridioides difficile infection. Some of those symptoms would be watery diarrhea, no appetite, that's anorexia, nausea, belly pain, and tenderness of the belly as well. Okay? Usually we diagnose Clostridioides by looking for toxins A and B. Toxin A makes you have all the watery diarrhea, while toxin B actually makes you have cells in mucosa in the diarrhea because it actually starts to destroy the intestinal mucosa. Like I said, the best way to help identify Clostridioides difficile is by identifying toxins A or toxins B, and we can do that by doing an ELISA test or some sort of molecular test. Okay, we can also attempt to grow out Clostridioides difficile on CCFA agar, but it can sometimes be difficult to grow because it is so fastidious. CCFA is selective for Clostridioides difficile, and that stands for cycloserine cefoxitin fructose agar. The influenza virus is an orthomoxyvirus, and from October of 2018 to May of 2019, this influenza virus caused 40 million cases of influenza throughout the world and 50,000 deaths, so it is significant. It is a respiratory disease that can be anywhere between mild to severely limiting. Usually some symptoms of influenza virus would be things like body aches, fever, sore throat, cough, things like that. Okay, it is considered a seasonal virus that occurs every year between October and May, and there's two main types of influenza viruses. Those are called type A and type B. Okay, influenza virus is spread by droplets and it is highly contagious. There is a flu shot though that prevents influenza virus. The composition of the flu shot changes annually to match the circulating flu virus every year. It's interesting to note that the flu shot also protects you from H1N1, which is the swine flu. There are about 3,000 to 49,000 deaths annually in the United States, and there's a couple of lab tests that we can do to help diagnose influenza virus. We can do a rapid kit test, also known as a rapid influenza diagnostic test, or we can do viral culture, which can take up to 14 days, so it's pretty slow, or we can use polymerase chain reaction, or PCR, which is a molecular way of identifying the influenza virus. Some interesting statistics from 2020. There was 1 week in 2020 where there were 576 flu hospitalizations, and out of those 576, about half of those were influenza A and half were influenza B. And that was in 1 week. Now, from September to July of 2020, there were actually 7,299 influenza cases in Nebraska. Avian influenza is also known as H5N1, but there's a new avian influenza out called H7N9. These are influenza A viruses that are known to infect birds. Now, occasionally humans can get avian influenza, but that is rarely isolated in humans. In fact, the person-to-person transmission percentage is quite low. This is a naturally occurring influenza virus among wild birds worldwide and can actually infect domestic poultry. And that's why it's so significant, because people that work in the domestic poultry industry may lose all of their birds. Okay. Anyway, some symptoms of avian influenza would be things like fever, cough, sore throat, muscle aches, and pneumonia, very similar to other influenza strains. The way that we can identify avian influenza is molecularly through PCR or viral culture. Okay. There was a case in Chicago several years ago where there were 200 birds in a Chicago forest preserve that died of avian influenza, and that was just last year. The CDC says is that this poses a low risk to people, and since 1997, only 880 people have actually had the disease. So really, that's not too many in the big scheme of things. And I just thought this cartoon was kind of witty. I'll try the turkey with a little chestnut and penicillin stuffing. Wah wah. Swine influenza, also known as H1N1, causes a respiratory virus in pigs and humans. The swine flu variant is what infects humans. This is also known as H1N1V, which is a subtype of influenza A. This is a virus most closely related to North American and Eurasian swine origin H1N1 influenza virus. It was first identified in Californians working closely with pigs and later found to be transmitted from person to person. Now, to diagnose swine influenza, we can use rapid tests, rapid influenza tests, or we can use molecular things such as PCR, viral culture, or direct or indirect fluorescent antibody testing. Okay? Swine flu cases worldwide through 2009. There were 89,921 worldwide and 33,902 in the U.S. And that was in 2009, kind of during the swine flu's heyday. That was before our current vaccines actually protect us from H1N1. Okay? Now, the cases for swine influenza have dropped significantly due to the vaccine. It was a slight spike in 2012, but it has decreased since then. Swine flu, caution, it's airborne. From an epidemiology standpoint, looking at the progression of swine flu is quite interesting. It really puts a positive spin on how our CDC can come up with a vaccine so quickly for a strain of influenza. So in May of 2009, the CDC discovers that swine flu is actually a thing, and it's a variant of the H1N1 virus, okay? It's actually a variant of 4 other influenza viruses, okay? In June of 2009, the CDC, so this is a month later, the CDC declares a pandemic, and 50% of all flu cases were H1N1. In September 2009, there was a vaccine that was approved by the FDA, so that's only 4 months later. In August of 2010, this is a year later, the World Health Organization declares the end of the H1N1 global pandemic. Now, that is largely in part because of the swine flu vaccine. By November of 2011, 83% of flu cases tested were influenza A and less than 1% were H1N1. 17% incidentally were influenza B. But just you can kind of take a look at the progression of the swine flu, and pretty much within a year, they already had a vaccine, and now they include that in your regular influenza vaccine. I actually had the swine flu in 2009, and it was unpleasant, to say the least. You don't want to get the vaccine. Let's talk about the Flaviviridae viruses, which would include West Nile virus and Zika virus. Okay. The leading cause of mosquito-borne diseases in the United States is West Nile virus. This is a seasonal virus that is spread by the Culex mosquito, and symptoms of West Nile virus would include rash, fever, swollen glands, stiff neck, and it could eventually lead to coma. The best way to identify West Nile virus is by doing serum or spinal fluid ELISA, looking for IgM antibodies. So far, there have been 68 cases in Nebraska up until 2015 and 2 deaths. There is no approved vaccine at this time, but clinical trials are taking place on humans. Okay. One thing that's interesting is there's a fever in some people that have West Nile virus, and 1 in 150 develop central nervous system symptoms. Okay. Zika virus, on the other hand, there's no local transmission reported in the U.S. in 2018, but it's actually spread by the Aedes mosquito. Some symptoms of the Zika virus are asymptomatic, which doesn't help us at all, or fever, rash, joint pain, conjunctivitis, and it can be passed in utero from mom to baby. I'm sure you remember hearing about this in 2019 when there were several cases of Zika virus in South America and Central America, and this is actually countries that are more at risk of Zika virus. Okay. Now, Zika can be transmitted from mom to baby. It can actually result in microencephaly for these children. The treatment of Zika virus would be rest, fluids, pain, and fever reduces. There's no vaccine currently to prevent Zika, and the lab tests for identifying the Zika virus would also be IgM antibody ELISA tests and real-time PCR. I want to talk just a little bit more about West Nile virus. Like I said, it is a Flaviviridae virus. It is a seasonal North American virus spread by mosquitoes who have taken a blood meal from an infected bird. In 2018, there were 79 cases reported in Nebraska, and symptoms are very typical of viral illnesses: rash, low-grade fever, nausea, swollen glands, stiff neck. Blindness is kind of one of the more serious signs and symptoms. Paralysis and coma are also two more serious symptoms of West Nile virus. The best way to identify West Nile virus is looking at serum or spinal fluid IgM specific antibodies, and you can do that by using the ELISA method. Lyme disease is actually transmitted the bite of a tick, a black-legged tick. The organism that causes Lyme disease is called Borrelia burgdorferi, and it will cause a person to have a headache, pretty severe headache, fatigue, chills, fever, but really the diagnostic way of telling your patient that they have Lyme disease is by looking for their characteristic rash that almost looks like a target. I'll show you a picture of that in a moment. Some complications of Lyme disease, if it's not treated well, if it's not treated early, it's going to lead to systemic joint damage, heart damage, and cerebral spinal fluid problems or central nervous system depression. A lot of these viral diseases or interesting bacterial diseases can be best diagnosed by using ELISA or indirect fluorescent antibody, and even Western blot. Now, you probably recall from previous lectures, Western blot is mostly used to confirm cases of HIV. However, in the case of Lyme disease, you have to have two markers that are positive in order to be diagnosed with the disease. So in other words, you have to have a positive ELISA and a positive I don't know where I was going with that. Okay, you have to have both the Western blot and an indirect fluorescent antibody test. Both of those have to be positive in order to diagnose Lyme disease. Now, other tests would be urine ELISA, bacterial culture, or lymphocyte assays. The CD57 cells, that's a marker to indicate our people might have Lyme disease. Okay. From 2000 to 2016, there were 132 confirmed cases of Lyme disease in Nebraska, and you are always going to see high percentages of this disease in northern climates like Minnesota, Wisconsin, and the coastal northeast region of the United States. Okay. Now, like I said, this is caused by the bacteria Borrelia burgdorferi. It is transmitted through black-legged ticks. Most cases can be treated with antibiotics, and the CDC recommends confirmation with Western blot testing. So here is the pronounced and very prolific rash associated with Lyme disease. I mean, I think if I had this target-like rash, I would be heading to the doctor and asking if I had Lyme disease. Very typical. Tuberculosis. This is kind of crazy, but a lot of people think it's been eradicated in this country, and that's actually not true. 1/3 of the world population has tuberculosis, and some people develop what's known as latent TB, where they have the bacteria but no symptoms, and they may develop it in the future. And they can be treated with antibiotics for this type of tuberculosis. Tuberculosis is definitely on the rise, and there's a new case diagnosed every second. And there's about 2 million people that die every year from the tuberculosis illness. Okay. Projected cases over the next 10 years, 1 billion people will be afflicted with the disease, and 36 million people will die from it in the next 10 years. Now, in the United States, the incidence of tuberculosis has been on the rise since the 1980s. Okay, the reason that it is on the rise is going to be covered in the next couple slides, but it's definitely on the rise. When I used to work at Bryan LGH West, it would be maybe once or twice a year we would see a positive tuberculosis case, but by the time I left, about 8 to 9 years later, we were seeing maybe 5 or 6 a month. So the cases of tuberculosis are definitely increasing. And then here is kind of a skinny little fellow that is being treated with oxygen and some medications to prevent any further destruction from tuberculosis. So like I said, Mycobacterium tuberculosis is on the incline in the United States, and there's a number of reasons why. First of all, federal funding is lacking for TB eradication. There is an HIV and AIDS epidemic that's been going on in our country since the 80s. Anytime you have immunosuppressed people, it is going to make them more prone to developing Mycobacterium tuberculosis. So that's a concern. We have immigration from endemic countries, so we have people coming over here that are already positive for tuberculosis because it is endemic to their country and they're bringing cases of TB into the country. Overcrowding is very much of a concern when people have Mycobacterium tuberculosis because it is spread by droplets. So if you're in a highly populated area and you have Mycobacterium tuberculosis, and you're coughing, you're going to cough those droplets onto your neighbor. Whereas one of the best things you can do is stay away from big crowds if you're trying not to get sick with something communicable. And then finally, the last reason why we see tuberculosis on the rise is there are a number of multi-drug resistant strains. I'm not sure how they're going to get around that other than giving two drugs at the same time that are synergistic to one another. That would help. There are some populations that are considered risk groups according to the Centers for Disease Control for getting Mycobacterium tuberculosis. If you're a person living in close contact with other infected parties, it's pretty communicable. You're likely to develop TB. If you're an HIV patient or if you're in some way immunosuppressed, you're going to be a lot more susceptible to HIV. Now, illicit drug users that are sharing needles and things like that, they're way more likely to develop Mycobacterium tuberculosis because they're not using safe habits. If sometimes you have employees in high-risk settings like prisons, shelters, even healthcare workers, these are all people that are at higher risk for developing tuberculosis. Then we have foreign-born U.S. residents from endemic countries. Again, tuberculosis is endemic in their country, and so when they bring it over here, we may feel like it's no big deal, but we certainly have to treat that person for tuberculosis and quarantine everybody that's come in contact with that person. We do want to keep it out of the U.S., all right? Low-income populations are more likely to experience Mycobacterium tuberculosis, mainly because they don't have access to good health care. And then finally, infants and children of the adults in all of the former risk groups. Here's kind of a nice map where you can see where populations of tuberculosis are more common. You can see that in Russia and in Africa, there is a high amount of tuberculosis that runs rampant in those countries. And then you take a look at on the left, the United States and Canada, where the cases of tuberculosis is much lower. Now, when we were looking at that last map, please understand that there is a vaccine known as the BCG vaccine. It is available in many countries, but not recommended generally in the United States. We just don't think it's a super effective vaccine, but there actually is one outside of the U.S. Now, keep in mind, tuberculosis is a rapidly spreading disease, and the specimen of choice is usually some sort of respiratory sample, like sputums or bronchwashes. However, you can have other types of mycobacteria that's going to cause tuberculosis-like symptoms, and you can see those other types of mycobacterium in spinal fluid, in the tissues, and in the urine. The best way to diagnose tuberculosis is by doing an acid-fast stain, and you can either do the Kinyan's carbofusion or the auramine-rhodamine, but the auramine-rhodamine is much more specific and sensitive. Don't forget, whenever we're processing acid-fast samples, we want to make sure that we digest them and decontaminate them. And we can do that by using N-acetyl-L-cysteine as the mycolytic agent that actually breaks down the tissues and the purulent material that you coughed up. It can actually break that down, releasing Mycobacterium tuberculosis onto the culture media so we can culture it and identify it. Now, 4% sodium hydroxide is the decontamination unit where we kill off normal flora in order to be able to see the tuberculosis organism growing underneath the normal flora. Like I mentioned, there's a couple stains for Mycobacterium tuberculosis, including the Kinya's carbofusion and the auramine rhodamine. It's usually the Kinya's carbofusion that we consider to be the confirmatory test, and the auramine rhodamine is the initial screening test for Mycobacterium tuberculosis. Now, there are some differences between these two staining processes. In the Kinya's carbofusion, this is considered a cold stain. And the reason it's called the cold stain is there used to be another test that used the microwave and warmed it up. It's not on this list, but I think it was the Zeal-Nielsen type of stain that used a tiny amount of heating. But we really don't use a lot of heating in Kinya's carbofusion or auramine rhodamine. Kinya carbofusion is considered a cold stain. The organisms will appear pink on a blue background. Okay. Now, the other stain is called auramine rhodamine. This is a fluorochrome or fluorescent staining test. It's much more sensitive than Kinya's carbofusion, and we are going to see orange organisms, bacillus, on a black background. Now, you may see chunks of orange that are debris or other things, but you really should see primarily the tuberculosis organism. If you're seeing artifacts, just ignore those, okay? Auramine rhodamine is a very sensitive stain, and we always want to view a minimum of 300 oil immersion fields. Okay, so anytime we're culturing for Mycobacterium tuberculosis, we want to use a media that is appropriate for a bug that grows very slowly. So one media we can put Mycobacterium tuberculosis on, or at least the inoculum, we can put the inoculum on what's known as a Lowenstein-Jensen media. A Lowenstein-Jensen media is a media that does not dry out very easily, and so it is a good media to support a bug that takes 3 weeks to grow, okay? So on this Lowenstein-Jensen media, you can see the rough and buff colonies of Mycobacterium tuberculosis. Another media that we might use for Mycobacterium tuberculosis is Middlebrook 7H11, which is a thin pored plate. We can actually take that thin pored plate and put it on the microscope stage and then observe colonies with our 10x and 40x objectives. Sometimes you're going to see tiny colonies that actually demonstrate that serpentine cording. That's pretty suggestive that the person has Mycobacterium tuberculosis. Now, one thing you might want to know is that patients that have tuberculosis are usually treated for 6 to 12 months, and usually they are treated with 2 different drugs to prevent resistance. All right, let's talk about MERS, Middle East Respiratory Syndrome. It was first recognized in Saudi Arabia in 2012, but has since spread to other countries. It's very low risk in the United States, but some speculate that it's actually associated with camels. It is a coronavirus. Symptoms of people that have MERS have respiratory symptoms, shortness of breath, fever, and cough, and 3 out of 10 will die of this syndrome. There is no vaccine currently available for MERS. However, it is similar to COVID-19, which is a severe acute respiratory virus or a SARS virus. Coronavirus 19 is responsible for 3.1 million doses of vaccine being developed, and that's how many vaccines were given in Nebraska in 2022. Like I said, there is no specific vaccine for MERS. MERS there's a very low risk of developing MERS in the United States. There have only actually been 2 cases, and those were both in 2014. There was 1 case in Indiana, and 1 case in Florida. Both were healthcare workers who had formerly worked in Saudi Arabia, so that's kind of interesting. The treatment of MERS is supportive care and protection of the vital organs. And the diagnosis for MERS is real-time PCR from respiratory secretions or serum. Let's talk a little bit about dengue, which is a Flaviviridae disease, another Flaviviridae disease. Dengue fever is something that is common in places like Puerto Rico, Dominican Republic, South America. There's usually approximately 50 to 400 million cases annually, and there are cases that have been reported in places in the United States like Florida and Texas. Like I said, it is endemic to the tropics and subtropics of South America, the Caribbean, India, Southeastern Asia. And this particular disease is spread by the Aedes species of mosquito. This is what we call an arbovirus, and an arbovirus is any virus that is spread by an insect. The Aedes mosquito takes the blood meal and becomes infectious, bites another human who then becomes infected with dengue fever, and rarely, very rarely, is transmitted through blood and organ donation, okay? And occasionally it can go from mother to baby, but again, that's very rare. This dengue fever is caused by a single-stranded RNA virus that's actually a blend of 4 flaviviride diseases called dengue virus 1, 2, 3, and 4. Dengue is the leading cause of death in the tropics and subtropics, places like the Dominican Republic and Haiti and Puerto Rico. As of 2018, there were 21 to 50 cases of dengue virus near Omaha, well, especially in Douglas County and Sarpy County, and there were 11 to 20 cases in Lincoln. Most cases that develop in the United States are in south central Arizona. Okay, it's the leading cause of death in the tropics and subtropics, and there's 100 million new cases every year. It's caused by 4 types of mosquito-transmitted viruses. The mosquito bites an infected person, then it bites an uninfected person, and now this uninfected person is infected. Some symptoms of dengue: high fever, headache, eye pain, muscle and joint pain, rash, bleeding nose and gums, and we diagnose dengue by doing lab tests such as ELISA or serum PCR. Let's take a look at that Aedes mosquito, shall we? Here's an Aedes mosquito. Usually the types of mosquito we have in Nebraska are called Culex mosquitoes, but this Aedes mosquito, I actually have seen this kind of a little bit west of here. I saw the Aedes, and they're very easy to spot because they have these white stripes on them. Like I said, some symptoms of dengue fever would include headache, fever, joint pain, rash, bleeding from the mucous membranes, and it could actually lead to what's known as hemorrhagic fever. There really is no specific medication used to treat dengue fever, but you do want to make sure that fluids are being replaced and that pain-relieving medications are being given. There's currently no vaccine available for the prevention of dengue, and lab verification is by testing for IgM or IgG antibodies using ELISA, real-time PCR, or plaque reduction and neutralization test, which is used to quantify a viral antibody. Here we take infected cells mixed with antibody in vitro and determine how much antibody it takes to reduce cell plaques. So it's kind of interesting, plaque reduction and neutralization test. Here's another infographic that talks a little bit about the hemorrhaging you're going to get with dengue fever, also dizziness, vomiting, stomach pains, and a weak rapid pulse. You can see in the bottom right image that the hemorrhagic process of kind of hemorrhaging capillaries of the eye is occurring in that young man on the bottom right. Yellow fever is a Flaviviridae virus as well. It causes jaundice, which is why it's called yellow fever. This is a disease found primarily in Africa, Central and South America. However, it used to be endemic to the U.S. as well. It was quite deadly in the late 1700s to the late 1800s. Okay. In fact, in 1793, there were 50,000 dead in Philadelphia. In 1853, there was about 8,000 dead in New Orleans. In 1855, there was 3,000 dead in Norfolk, Virginia. And in 1878, there were 20,000 dead in the lower Mississippi Valley, and those were all deaths that occurred because of yellow fever. Now, there could be up to 170,000 deaths or cases and 60,000 deaths per year in Africa alone. It has a complex cycle of transmission where you have an insect vector, which is the Aedes mosquito, or hemorrhagic mosquitoes, which bite monkeys and transmit the disease to and from other primates, including the occasional human, which may take their infected cells to an urban area and transmit it quickly to others. But again, this is spread by a mosquito known as the Aedes aegypti mosquito. Let's talk a little bit more about yellow fever in the next slide. Some of the symptoms of yellow fever would be, first of all, you could be asymptomatic, but you may also have fever, chills, headache, backache, shock, organ failure, and death. You could be very severely jaundiced, as you can see in that eyeball on the upper right. The symptoms typically improve around 5 days, and 15% of people relapse and get liver disease and jaundice because of this illness. They treat yellow fever with rest, pushing fluids, giving them pain-relieving medication, and close observation. Okay? And there is a vaccine available, and it's typically recommended to people living in Africa or South America. There is a vaccine, and it provides lifelong protection, and there's no need for a booster shot, so that's kind of sweet. Laboratory verification, we can identify yellow fever by looking for IgM antibodies in patient serum or viral culture. And again, viral cultures are kind of slow and a pain in the butt. So if you can identify it using ELISA, that would be even better. Now, Crosse encephalitis, you may have heard of this because this does occur mostly in the Midwest, Mid-Atlantic region, and the southeastern states. The hosts for Crosse encephalitis are chipmunks and squirrels. Humans will get sick but not have enough virus to infect the next biting mosquito, so we're kind of what's known as a dead-end host. Crosse encephalitis is the cause of 62% of all central nervous system infections, especially in the Midwest. It is an arbovirus that's transmitted by the Aedes mosquito. Symptoms of Crosse encephalitis would be mild fever, headache, nausea, central nervous system depression, and usually when that occurs, it's now become a much more serious disease. The treatment is not great, it's just supportive care, and the diagnosis, again, is either serum or spinal fluid, IgM, ELISA, antibodies. This is one of my favorite diseases to say. This is Chikungunya fever. This is a Tobeviridae virus. It is an arbovirus, so it is transmitted by yet a mosquito again. There's actually several species of mosquitoes that transmit Chikungunya virus. The symptoms would be fever and joint pain, and there's no vaccine available. Okay, the diagnosis is looking for serum IgM antibodies or some sort of molecular testing like nucleic acid testing. And the treatment, like a lot of viral diseases, is simply supportive care. This is a disease seen in the United States, also South America, India, and Southern Africa. Now, there were 90 cases in 2018 to 2019, and all cases didn't originate in the U.S. They were actually diagnosed in travelers. Now, none of these 90 cases were found or diagnosed in Nebraska. Borrelia miyamotoi is an arbovirus transmitted to humans via the hard-bodied ticks. The infected organism is transmitted by an insect, and that's why it's called an arbovirus. It is a spiral-shaped bacteria that closely is related to one that causes tick-borne relapsing fever. Borrelia is a spirochete, and the symptoms of this disease would be things like fever, chills, headache, joint pain, and fatigue, very similar to a lot of other arboviruses. There's no such rash that is seen, seen with Borrelia as there is one with Lyme disease. The diagnosis is PCR or antibody detection, and the treatment is doxycycline. Usually they treat you for about 2 to 3 weeks for this disease, amoxicillin or ceftriaxone. Let's take a look at Borrelia in an image on the next slide. This will make your skin crawl. Here you go. So if you thought microfilaria are bad, how about having some spirochetes in your blood? This is Borrelia, and you can see those spiral-looking organisms in the peripheral blood smear. And you can see they're pretty lengthy, probably about 40 microns in length. Onchoceriasis is a disease known as river blindness. It's the second leading cause of blindness in the world, and it affects about 25 million people according to the World Health Organization. 800,000 of these have visual impairments. In 30 countries it is seen in, like Africa, Yemen, and the Middle East. This is caused by the filarial worm Onchocerca volvulus, and it is vectored by the black fly Simulium species. Symptoms would be blindness, skin nodules, and itching. I've been itching like crazy. I hope this is not Onchocercus. But anyway, I digress. This is seen worldwide. It's the second leading cause of infectious blindness. I already mentioned that. The diagnosis is skin biopsy or PCR. If there is no filaria seen, that's when you want to do PCR. You might actually see filaria on the peripheral blood smear. Treatment would be things like ivermectin, and that would be, you would be on it for the rest of your life to prevent microfilaria. There is no vaccine, unfortunately, and there are no prophylactic medications. Okay. All right. So that's onchoceriasis. That's a tough one to say. Let's talk about Q fever. Well, before we get to Q fever, let's take a look at a nice infographic about river blindness. I'm just going to give you a minute here to take a look at the life cycle of onchoceriasis. Rocky Mountain spotted fever is one of the deadliest tick-borne diseases in the U.S. It is caused by a rickettsial organism known as Rickettsia rickettsii. It is a febrile disease, so it is associated with a high fever, and it is common in the United States. It is vectored by the American dog tick, and the symptoms include fever, headache, nausea, and rash. And we can diagnose this serologically by using the febrile agglutination test, where Proteus vulgaris OX19 antisera is mixed with the patient's serum, and if it clumps, it means that our patient has a febrile disease with a rickettsial organism. You can also use indirect fluorescent antibody. The treatment of Rocky Mountain spotted fever would be doxycycline or chloramphenicol. Okay. Now, if there's a delay in treatment, this can result in death. Always want to treat based on history, if they have a tick bite or what their travel history is. Some more harsh symptoms, not only is this a fever-producing disease, and they'll have headache and nausea, but these patients may also have muscle pain and vomiting. Let's take a look at the American dog tick. Ew, this is an American dog tick. Yucky. Q fever is named for query because they didn't know where it came from in the beginning. It naturally affects goats, sheep, and cattle, and people get infected by breathing dust from animals, feces, milk, and urine. There are about 180 cases every year, and 68% of the people that have Q fever end up hospitalized. This is caused by a gram-negative intracellular bacteria known as Coxiella burnetii. It can infect animal placenta, amniotic fluid, urine, feces, and milk. People are infected by breathing the dust particles of any of those above substances, and the symptoms are very similar to other rickettsial diseases, kind of flu-like, chills, fever, fatigue, under the weather, muscle pain. And the diagnosis of Q fever, again, is febrile agglutination test, indirect fluorescent antibody, complement fixation, or something molecular like PCR. The treatment is doxycycline or hydrochloroquine. Human Ehrlichiosis is a tick-borne intracellular bacteria. So it is spread by E. chafensis or E. ewingii. Those are tough ones to say. The vector for this disease is a Lone Star tick. Symptoms would be fever, headache, fatigue, muscle aches, and the diagnosis would be PCR or IFA, indirect fluorescent antibody. And microcolonies or Ehrlichia may be seen in peripheral blood smears, and the treatment, of course, is going to be doxycycline. So let me take you to the next slide where you can see a colony of Ehrlichia inside of a white blood cell. Here's an example of what Ehrlichia looks like in a white cell in a peripheral blood smear. I'm not sure that I would be able to say that's Ehrlichia, but I would certainly recognize it as being something that is probably bad. Trench fever, cat scratch disease, and carrion's disease are all carried by sandflies. Trench fever is caused from a gram-negative intracellular parasite known as Bartonella quintana. Other Bartonella species cause cat scratch fever. I almost sang that, cat scratch fever. It is vectored by the human body louse, and some symptoms of trench fever would be fever, headache, rash, severe bone pain in the shins, neck, and back. Okay? This was associated with cat fleas, and we want to make sure that to prevent the spread of trench fever, people aren't sharing bedding, clothes, and towels. Okay? Another symptom of trench fever is that you can develop what's known as memory problems. Trench fever was something big in World War I, especially when there were guys in the trenches and they would spread human body lice back and forth from one another, and those body lice would carry Bartonella. And so the soldiers would end up getting what's known as trench fever. Okay. You've probably heard some interesting catchphrases in popular culture that have came from the word louse and lice. For example, stop nitpicking me about how lousy I feel. Sheesh. That obviously comes from, it's associated with the human body louse. Okay. German measles are also known as rubella or the 3-day measles. This is a paramoxavirus. It's also known as rubella. Red measles, which is much worse, is rubeola, and it can affect the lungs. German measles is spread by coughing or sneezing and is quite contagious. Okay. Some symptoms: fever, runny nose, cough, red eyes, sore throat, and rash. And babies can be born with congenital measles, and it's very serious for them. Or mom can actually have a miscarriage of her baby because she has German measles. This is diagnosed through ELISA, RNA detection, or genetic analysis, such as molecular epidemiology. There is a vaccine for it called the MMR vaccine, the measles, mumps, and rubella shot, and the disease is on the incline. It is on the incline because people are not getting their vaccines or allowing their children to get the vaccines like they probably should. This is spread by person-to-person droplet contact, and it is endemic in Africa, Middle East, Asia, and there are about 100,000 infants every year that are born with congenital rubella. Mumps is a paramoxavirus as well. It is highly contagious. Symptoms would be low-grade fever, muscle aches, malaise, no appetite, swollen salivary glands, and that's known as parotitis. Even with a vaccine, there are cases of mumps that still occur. You get it from close contact with other people that have mumps, and you can even get it sometimes with the vaccine. And cases are on the incline for mumps as well. Now this one sounds very ominous. This is devil's grip disease or Bornholm disease, also known as pleurodynia, named after the Danish island of Bornholm. Okay, this is a picornaviride virus or a Coxsackie virus, and the symptoms would be pain in the chest or abdomen, pleurisy, headache, sore throat, blisters of the mouth, hands, and feet. It is self-limiting. Usually within a week or so you're starting to feel better. Sometimes there are outbreaks in schools and nurseries because it is spread by contact with secretions, nose and mouth secretions, and also feces. Okay. There is no vaccine available for devil's grip. However, there is a treatment, and that treatment would be steroids. Rift Valley fever is mostly seen in cattle and goats, but can be transmitted to humans. It was first reported in Kenya's Rift Valley, and that's why it's called Rift Valley fever. Epizootic outbreaks can result in increased animal deaths and economic losses for the country this occurs in. Okay, it's a febrile disease common in domestic animals, and the symptoms of Rift Valley fever would be low-grade fever, weakness, dizziness, and back pain. 8 to 10% of infected people develop an eye disease, and they will actually have lesions of the eye. And about 1% or a little bit less than 1% can develop hemorrhagic fever. This is diagnosed through ELISA or PCR, and about the only thing they can do for you if you have Rift Valley fever is support you with IV fluids and pain medications. Helicobacteriosis is an infection of the gastrointestinal tract with Helicobacter pylori. It causes inflammation of the stomach lining. Some symptoms of helicobacteriosis is inflammation, gastritis, ulcers, and stomach injury, including stomach cancer. It's a gram-negative spirochete and produces the urease enzyme and cytotoxin VAC-A protein. Children living in unsanitary or overcrowded conditions are definitely more prone to getting helicobacteriosis. To diagnose helicobacteriosis, what we want to do is an endoscopy or a breath test using looking for the compound urea. We can also look for the VAC-A protein, a protein that is a cytotoxin and forms it forms anion channels in target membranes, destroying cellular mitochondria. Okay. The treatment for helicobacteriosis is amoxicillin, clarithromycin, metronidazole, and tetracycline. Let's take a look at a picture of Helicobacter. Here's a creepy looking feller for you. This is Helicobacter pylori. Okay. Let's take a look at Ebola now. The Ebola hemorrhagic fever is transmitted from close contact or sexually transmitted. It is a deadly disease of people and non-human primates, mostly contained to sub-Saharan Africa, but people do get sick by coming in contact with an infected bat, primate, or dead body. Okay, a good test to diagnose Ebola is the BioFire film array molecular real-time amplification PCR test. Okay, symptoms of Ebola. It can be deadly in a very short amount of time, of course. Fever, gastrointestinal and respiratory collapse, and unexplained hemorrhages. Diagnosis, we already mentioned that, nucleic acid test, cell culture, or antibody detection. And sample sent to a laboratory response center must follow strict and specific guidelines because it is so communicable. In March 2016, you may remember this, there were 13 cases and 1,200 contacts in Guinea and Liberia. Most of those originated from West Africa. There is, unfortunately, no vaccine available for Ebola. One thing that's interesting about Ebola is that usually pandemics and epidemics don't get too far with Ebola because the virus spreads so quickly and people die so quickly that they don't have enough time to spread it around. Let's take a look at the picture of the Ebola virus. Here's the Ebola virus, and there's a link that you can copy and paste into your browser to see a little bit about the BioFire film array molecular real-time amplification PCR assay. This brings us to our last horrifying disease, which is variant Creutzfeldt-Jakob disease. This is the same prion responsible for mad cow disease in cattle, which is known in cattle as bovine spongiform encephalopathy. Okay, this is a human prion disease. A prion is an infectious agent composed of a misshaped protein, not that of DNA or RNA. This same prion, like I said, causes mad cow disease, and it causes what's known as a progressive neurodegenerative disorder. It is fatal, okay? Symptoms would be dementia or other neurological signs, and there are about 3 cases for every million people. So it's quite rare. There have been 4 cases reported in the U.S., and those people all had strong ties to the United Kingdom and Saudi Arabia. Variant Creutzfeldt-Jakob disease is not transmitted from person to person. It has been implicated in the use of blood products, and lab tests for variant CJD would be an autopsy or suspicious vacuoles in the brain tissue, maybe a neuropathic exam of the brain tissue. Those are all ways to diagnose variant Creutzfeldt-Jakob disease. Unfortunately, it's usually diagnosed post-mortem. So that is it for the hot topics in infectious diseases. Today we talked about all kinds of things, including Clostridioides difficile, a little bit about avian influenza and swine influenza, a little bit about West Nile and Zika and Lyme disease, talked a bit about tuberculosis, Middle Eastern respiratory syndrome, dengue virus, yellow fever, Onchoceriasis, and human Ehrlichiosis, German measles, and mumps. Okay, so that's what we talked about today. I hope you enjoyed it. You probably learned a whole lot. This was a little bit longer of a lecture than I was hoping for, but hey, it was still fun. You learned a lot. Have a good day. Bye.